Hello, welcome back. We are going to start on our first approach into cardiac physiology, taking a look at the heartbeat and the electrocardiogram, EKG, ECG, either one, they're both the same. EKG is more German rooted because the cardio starts with a K. All right, so before we get into the actual details of the electrical activities of the heart, just an overview of some of the physiology things we're going to take a look at. So we start with the electrical stimulus at the SA node, which is going to lead us to the distribution of the conduction system and the ECG. So this video is going to cover those things. The second video, because the electrical activity will trigger cardiac contraction, we are going to take a look at the cardiac cycle and the phases of that, so diastole and systole and pressures and volumes and things like that. And then we're going to take a look at cardiodynamics, which is, it's not listed here, but it's going to be all the factors that influence the function of cardiac contraction and blood flow. Okay, so when we take a look at that signal that starts all of the muscle contraction, it starts at a region of the heart called the SA node. It is embedded at kind of the upper right-hand corner of your right atrium, and it stands for sinoatrial node. The SA node, sometimes called the pacemaker because it's the one that sets the pace. It is not nervous tissue. It's not like a neuron. It is cardiac muscle tissue that has been modified to generate these unique electrical signals. So don't think that it's like neurons. It's not nervous system. It's muscular system, but we know muscle tissue can be excitable. They have that voltage gated channel so they can send these signals, and that's exactly what happens. So in the SA node, we have a characteristic, well, let me back up. The whole heart itself has characteristics of what we call prepotential or spontaneous depolarization. It's just the SA node reaches that threshold faster than any other part of the body, any other part of the heart, sorry. So before we jump into describing what prepotential is, I just want to take a quick memory, walk down memory lane of a typical action potential, right? So we have -70, -60, 0, +30. This is not to scale, clearly. So let's say we're at rest, right? So at rest, -70 millivolts. Oops. Here we go. -70 millivolts, a graded potential comes in, chemically gated channels open, sodium rushes in, depolarizes the cell to threshold, right? So we're depolarizing to threshold. At threshold, at the initial segment of the axon of a neuron, all those voltage gated sodium channels open, sodium rushes in, we're in rapid depolarization. +30, sodium channels closed, potassium channels open, we come back, rapid repolarization. Channels close, get inactivated or back to resting state. We do a little bit of hyperpolarization. Sodium potassium pump gets us back to rest, and we stay at rest until another stimulus comes in. So if a stimulus shows up like right here, another stimulus shows up, and then we will reach threshold, another graded potential. So all of that is pretty much the same with the physiology of what's happening at the SA node. Here are some differences. One, you'll notice the values are a little bit different. Okay, threshold in this case is at -40 millivolts instead of -60, and resting is closer to -60 than it is at -70. But those are just numbers. The physiology is all still the same. So with these cells in the SA node, the AV node, the rest of the conduction system, and even the heart muscle themselves, they have a unique property in that they cannot maintain resting potential. So down here, right, we can maintain resting potential, and we could maintain it all the way forever until a graded potential comes along. Well, cardiac muscle tissue can't do that. Let's see, we'll track, I use the dark blue. So let's see, this is our action potential. We track down to rest. Now in a normal neuron, this would stay at rest, right, this blue line I'm drawing, until a graded potential comes along. Well, they can't do that. Cardiac muscle cells, and especially in these SA node, they do their action potential, depolarization, repolarization, back to rest. But take a look at what happens. They spontaneously depolarize, which means there's probably some leak channels going on. So it cannot maintain the resting state at -60, So it slowly leaks back up to threshold without any other incoming information. And then once it reaches threshold, it does the same thing. Voltage gated channels are open, closed, open, back to rest, but I can't maintain rest, so I leak up, and then I hit threshold, and then I'm going to do it again, and so forth and so on, and that sets the pacemaker cells. So the reason why the SA node is the pacemaker is because it does this spontaneous depolarization, trying to like this, faster than any other part of the cell. So if this is my SA node and my AV node is below it, AV node is more like this. It does the same thing, but its pace is slower. This is going to trigger this to fire. And then it will trigger this to fire. And every time the SA node fires, the AV node is going to fire because it is getting stimulated from the SA node. If we took the SA node away, the AV node rate would just be a little bit lower. And I think there's some statistics in the book. I don't have it written down. I think the SA node is like 100 beats per minute all by itself. The AV node might be like 80 or 60 beats per minute. The bundle might be 40 beats per minute. So the rate of the spontaneous depolarization decreases the further away you get from the SA node. But it doesn't matter because the SA node is the king. It's the one that like tells everybody else what to do. So that prepotential is really important in the triggering of a heartbeat, and it is independent of any nervous input or endocrine input to initiate it. Those inputs can change it, right? So think sympathetic activation can speed it up, parasympathetic can slow it down, hormones like epinephrine and norepinephrine, they can change heart rate, but they cannot trigger or initiate the heartbeat itself. That comes all by itself in the heart, and there is a term called automaticity. Automaticity is the ability of a cell to reach threshold and do an action potential all by itself outside of any other input. So let's take a look at some of the other parts of this, what we call the intrinsic conduction system. So we have the SA node starts a signal, communicates to the AV node via these internodal pathways, just little branches of these conducting cells. Then we have the next big chunk pieces called the AV node. AV node goes into what's called the AV bundle, kind of heading towards the atrioventricular septum. It splits then into your left and right bundle branches. And then towards the apex of the heart, the bundle branches kind of turn up, kind of a 180, and go back up and distribute in the walls of the ventricles. And these are called your Purkinje fibers. Okay. So other than the internodal pathway, there's 5 major parts of this intrinsic conduction system: the SA node, the AV bundle, the bundle branches, and the Purkinje fibers. Make sure you know those and you know them in their proper order. All right, so here we're going to take a look at kind of the steps and the timing associated with the intrinsic conduction system, kind of starting at time 0, right? So time 0, SA node fires, atrial activity begins. In about 50 milliseconds, it takes to travel that message from the SA node to the AV node. And then at that AV node, there's what we call a 100 millisecond delay. So the signal goes from the SA node to the AV node. The electrical activity travels a lot faster than the actual contraction. So the signal travels, then the contraction kind of follows along. And we have this 100 millisecond delay at the AV node because what that does is it allows for all the atria to contract before we start telling the ventricles to contract. Because if we were to contract the atria and the ventricles at the same time, the blood would be pushing against each other and it's not going to be very efficient. So we want the atria to contract first, get all of that done, then the ventricles are going to contract. So it is coordinating these two atria, ventricles. We don't want them contracting at the same time. So that 100 millisecond delay is just a little hangout spot at the AV node, waiting for the atria to contract. Then it starts sending its signal down the AV bundle and the bundle branches. So it takes about 25 milliseconds to get down the septum. Okay, and then it turns the corner, goes up the Purkinje fibers, and now this signal, this electrical signal, is being sent to all the musculature of the ventricles, and then ventricular contraction can begin. So about 50 milliseconds for that signal to dissipate through or distribute through the musculature, and then the musculature responds by contracting. Okay, so that's not very much time. 500 milliseconds is a half of a second. So we're talking about a quarter of a second's time, which is even hard for our brains to comprehend, is all it takes for this electrical activity to happen, and then the muscle contractions will follow this electrical activity. Okay. We have been able to figure out, we humans, scientific community, have been able to figure out a way to record this electrical activity that's happening in the heart muscle itself, and it's called the electrocardiogram. So the let me get a brighter color, ECG, or K, if you like the EKG, either one's fine. So the ECG is a recording of the electrical events of the heart, and we have this activity. I think we'll do this in lab, pretty sure. That should be safe in COVID days. So here we have some key waves of the EKG, and they're named by letters. So we have what's called the P, the QRS complex, and the T wave. Those are the main things I want you to take away from this as far as labeling. So the P wave represents atria depolarizing. It's just the electrical events of all the ions flowing, the depolarization, right? So becoming more positive, ions flowing in. So the depolarization of the atria is recorded as this first little blip. It's called the P wave. Then we have the QRS complex, or the QRS interval, which is representing the ion flowing or the depolarization of the ventricle musculature. Now, ventricles are way bigger than atria, so of course their electrical signal is going to be appearing a lot larger than that. And then the T wave is ventricles are repolarizing. So again, ions flowing, but flowing back to the resting state. So we have P, atria depolarization, QRS is ventricle depolarization, and T is ventricle repolarization. Now you might be saying, well, we're missing something. Where's atria's repolarizing? Do they repolarize? Well, yes, they do, but atria are repolarizing at the exact same time the ventricles are depolarizing, and the ventricles depolarizing just overshadows any of that electrical event of the atria repolarizing. So we just don't have any representation of that on the EKG. So you need to know if I were to draw a EKG, P, QRS, T wave, right? Practice drawing EKG and make sure you can label all of its parts and pieces. So this would be the P, Q, R, S, and T wave. Now, each one of these represents one cardiac cycle, or the initiation of one cardiac cycle. So in this particular recording, we have 4 heartbeats in this particular time. Now we have this pause here because there's a break. We have this break time between the end of one heartbeat and the beginning of the other heartbeat, because our heart isn't beating one right after each other. There's some limitations for the heart to be able to do that. But if you were to shorten that space between the T wave of one and the P wave of the next, then you can infer that the heart rate is increasing. If you increase the length between heart rate is decreasing. So hopefully that makes sense. Also, the sizes of these waves can be indicative of size of the heart or amount of ions flowing. And also these intervals, I'm not going to be too concerned about the intervals here, like the ST interval, the QT interval, the PR interval. But just do know that in higher level interpreting of ECGs, that those can be indicative of problems of different parts of that conduction system or ion imbalances. Another part of your article or case study assignment this term is electrolytes and how their levels can affect the appearance of ECG and how that can be interpreted as diagnostic of some kind of problem. The last slide in this video will be some examples of these irregular ECGs. So let's take a look. You don't need to memorize these. It's just kind of interesting to see how problems with the heart can be recognized on ECG. They're just interesting. So the first one is called premature atrial contractions, called PACs. They can be normal. They're just every once in a while. It just is like an extra, like a little blip. you get 2 P waves before you actually get the QRS. So premature, so you're having a regular heartbeat and it's just like a double digit before you get your ventricles contracting. Paroxysmal atrial tachycardia is premature atrial contraction triggers a flurry of atrial activity, but the ventricles are still able to keep pace. So you have your regular rate, and then you have a whole bunch of your SA nodes just firing, and your atria is firing, and then your ventricles are firing right after it. And then the last thing to deal with atrial problem is called atrial fibrillation, sometimes called AFib or AF. So if we take a look, we have our ventricles. They're kind of doing their thing. We've got a nice QRS. We might even see a P wave there, but we don't see any clear P wave. Fibrillation is like this. It's just kind of twitchy. There's no pattern to it. So atrial fibrillation is, it's just the muscles in the atria just kind of flutter and they don't pump. Now AFib is not as lethal as ventricular fibrillation because for the most part, your ventricles are passively filling anyways, and we'll see this in the cardiodynamics video. When the atria contract, they only contract about 20 to 30% of the blood into the ventricles, which means 70 to 80% of the blood enter without any help of the atria anyways. But there are some other negative effects to AFib that can cause other problems like extra, clotting, and which could be dangerous as well. The next one we have is premature ventricular contractions, PVCs. Again, this can be normal every once in a while. It's just like an extra, again, like the premature atrial contractions, a little blip, an extra QRS complex without any P wave, without any atrial activity triggering it. When you get a whole bunch of those, then that might be something you can go and take a look at your doctor, talk to your doctor about. And then we have ventricular tachycardia. Ventricular tachycardia. Tachycardia is a whole bunch of beats, like a faster heart rate. So this is your ventricle is just contracting without any atria activity happening prior to it. So this is VTAC, right? We've heard that maybe on like ER shows. And then the last one, this is a medical emergency. Ventricular fibrillation, VFib. So this is also known as cardiac arrest because your heart is not pumping. Remember, fibrillation is squiggly. If it's just jiggly and fluttery like this, there's no blood is being pumped anywhere, and that's an emergency. So when they do the clear shock with the defibrillator, what you're trying to do is get rid of the fibrillation. You want to get back that regular rhythm, that what's called sinus rhythm, to get your SA node firing, to your AV node, to your ventricles. You're using external electricity to try to restart that whole pacemaker of the SA node. And if you can't do that, then the person's at risk of dying because they cannot survive without a functional pumping heart. Okay, so that is our discussion of the ECG and the electrical activity, the electrical events and the physiology of the heart. Next, we are going to take a look at contractile cell activity and cardiac cycle, and that will be our second video on cardiac physiology. See you next time. Bye.